Hello students, this is Dr. Faizan Mirza. We are discussing how glucose is broken down gradually to release energy in respiration. So respiration. What's respiration? Energy from complex organic molecules are transferred to ATP. And this transfer of energy to ATP occurs along a series of reactions. The first of these is glycolysis. This is followed by link reaction, which eventually is followed by Krebs cycle, which leads to oxidative phosphorylation finally. These stages are involved in respiration when all respiration is occurring through aerobic pathway. Glycolysis can occur aerobic as well as in an anaerobic condition, but for link reaction, Krebs cycle and oxidative phosphorylation to occur, oxygen is mandatory. Glycolysis occurs in cytoplasm, link reaction and Krebs cycle occur in the matrix of mitochondria, and oxidative phosphorylation occurs in the inner membrane, which is the cristae of mitochondria. Glycolysis and Krebs cycle ends up producing ATP by the process of substrate level phosphorylation, which already has been covered in the previous video. Glycolysis, the net gain of ATP is 2 per glucose molecule. Actually, 4 molecules of ATP are produced, but 2 are used up, so the net gain is of 2 ATPs. But overall, 4 ATPs were produced per glucose molecule in glycolysis in the cytoplasm of a cell. In link reaction, no ATP is produced. In Krebs cycle, there are 2 more ATPs being produced by substrate level phosphorylation, but only in aerobic conditions. Oxidative phosphorylation ends up producing 28 more ATPs. Again, oxygen is mandatory for this process. So if you combine all the amount of ATPs, the total quantity which is produced here, so 2 plus 0 plus 2 plus 28, that's 32 ATPs which are available for the cell. If net gain ATP are not included and we include actual 4 ATPs which are produced, so it's 4 plus 0 plus 2 plus 28. So a total of 34 ATPs are produced in respiring a single glucose molecule. So that's the amount of energy that one glucose molecule can yield. What happens in glycolysis? Glycolysis occurs in cytoplasm of the cell. Glycolysis refers to glucose being split. Lysis means the splitting. So when glucose is split, it is split. I'm starting this cycle, this series of reaction, not cycle, this series of reaction from hexose. Hexose is a C6H12O6 compound. When this reaction is occurring, we see that hexose, which is a 6-carbon compound, turns into hexose phosphate and then hexose bisphosphate using 2 ATPs. So a phosphate group is added to hexose and another phosphate is added to a hexose phosphate to form hexose bisphosphate. The addition of these two phosphates is called phosphorylation. This is exactly where 2 ATPs get used up even before ATPs could be produced. This provides activation energy for glucose to react. The moment hexose bisphosphate is produced, this hexose bisphosphate starts to split into 2 molecules of triose phosphate, which are 3 carbon each. The triose phosphate molecules then turn into intermediates, producing 2 more ATPs by the process of substrate level phosphorylation. 2 hydrogens, not H+, 2 hydrogens are removed from here and they end up reducing to NADs. Intermediates are further converted to molecules of pyruvate. Pyruvate is a 3-carbon compound, and during this, another 2 molecules of ATP are produced by substrate level phosphorylation. So this is how the description goes of glycolysis. So initially you have phosphorylation occurring of glucose to hexose bisphosphate using 2 ATPs, then splitting of hexose bisphosphate into 2 triose phosphate molecules, and then oxidation of that triose phosphate to 2 molecules of pyruvate. 2 ATPs have been used up, 4 ATPs are produced, there's a net gain of 2 ATPs, 2 molecules of pyruvate are produced, and 2 reduced NADs have been produced here. All these compounds are in the cytoplasm yet, but if oxygen is available, they will enter mitochondrial matrix. So 2 molecules of pyruvate and 2 reduced NAD, they both will go into the matrix of mitochondria upon availability of oxygen. And all this series of reaction was occurring in the cytoplasm only. So these reduced NAD initially were in cytoplasm, but upon oxygen availability, they will go into the mitochondria. I hope that makes sense. This is followed by link reaction. What's happening in link reaction? We follow link reaction together with Krebs cycle. So what's happening in link reaction? In link reaction, we start with pyruvate. A single pyruvate molecule turns into acetyl, a 2-carbon compound, by losing carbon dioxide and a hydrogen. This is called as decarboxylation and dehydrogenation. Decarboxylase and dehydrogenase enzymes are required for this reaction to occur. Acetyl, which is a 2-carbon compound, adds with coenzyme A to form acetyl-CoA. CoA is a coenzyme A, already discussed previously. It is a carrier of 2 carbon units, which are acetyls. The pyruvate entered the mitochondrial matrix by active transport from the cytoplasm of the cell. Carbon dioxide diffuses out from the mitochondrial matrix from the cell and it is exhaled out eventually. The overall link reaction is pyruvate combined with coenzyme A and NAD to form acetyl-CoA and carbon dioxide and reduce NAD. Since link reaction is occurring once for one pyruvate, so it will occur twice for two pyruvates, which actually are coming from one glucose molecules. So we multiply the whole reaction by 2. So one pyruvate and end up producing these compounds. So if you multiply this by 2, so overall product for one glucose molecule ends up being that 2 carbon dioxide molecules are produced, 2 reduce NAD, 2 acetyl-CoA, no ATP used up, no ATP produced. This all occurs in the matrix of mitochondria. And coenzyme A, as already discussed, is just a carrier of acetyl group. And all these compounds are still in the matrix of the mitochondria. Then comes the Krebs cycle. What happens in Krebs cycle? Acetyl-CoA enters the Krebs cycle and CoA delivers acetyl to the Krebs cycle. In the Krebs cycle, matrix, which occurs in the matrix of mitochondria, the compound oxaloacetate, which is a 4-carbon compound, it's already present in the matrix of mitochondria. So when coenzyme A delivers acetyl to this, acetyl was a 2-carbon unit. When it combined with oxaloacetate, which is a 4-carbon compound, citrate 6-carbon compound is formed. This 6-carbon compound citrate undergoes decarboxylation and dehydrogenation. A carbon dioxide is removed and a reduced NAD is produced. The 5-carbon compound undergoes another decarboxylation and another dehydrogenation. The 4-carbon compound is formed, and the 4-carbon compound then produces ATP by substrate level phosphorylation and is then again undergoing twice dehydrogenations, once producing reduced NAD and once producing reduced FAD to once again form oxaloacetate, so the Krebs cycle can take another turn. So overall, acetyl, which comes from the Krebs cycle, can also come from fatty acid tails as well. So Krebs cycle can begin either from acetyl, which is coming from link reaction through the pathway where glycolysis was involved and pyruvate was turned into acetyl, or this can come from fatty acid tails. We know they are hydrocarbon tails, and hydrocarbon tails can be cut into 2 carbon small units to produce acetyl. Acetyl, as already discussed, is combined with oxaloacetate to form citrate, and it undergoes decarboxylation, dehydrogenation, as already discussed. Again, decarboxylase and dehydrogenase enzyme will be required here to carry out these reactions. Molecular rearrangement of the 4-carbon compound ends up producing ATP by the process of substrate level phosphorylation. And again, the Krebs cycle occurs in the mitochondrial matrix. And since we start off from one glucose molecule, we know that from one glucose molecule there were 2 pyruvates being produced and 2 link reactions occurred. So acetyl produced were also 2. So this Krebs cycle is just for one acetyl. So we know that for one glucose, since there were 2 link reactions occurring, there will be 2 Krebs cycles occurring, one for each acetyl-CoA that came from the breakdown of glucose in glycolysis. So what happened in Krebs cycle? If we try to summarize for what went on. So for one acetyl, 3 reduced NAD, 1 reduced FAD, 1 ATP, 2 carbon dioxide are produced. But since we are discussing for one glucose molecule, so we'll just multiply this by 2. So 6 reduce NAD, 2 reduce FAD, 4 ATP, 4 carbon dioxide and 2 ATP are produced. This was your glucose molecule C6H12O6. We know 2 carbon dioxide were removed in link reaction, 4 carbon dioxide were removed in the Krebs cycle. So this makes up all 6 carbon dioxide which are exhaled out for one glucose molecule in respiration. We know C6H12O6 gives 6 CO2 plus 6 gives 6 CO2 plus 6 water molecules and 2870 kilojoule of energy is produced. What about hydrogen carriers? We know one hydrogen carrier is carrying hydrogens. So 8 reduced hydrogen carriers were produced in Krebs cycle. 2 reduced hydrogen carriers were produced in link reaction. And 2 reduced hydrogen carriers were produced in glycolysis. A total of 12 reduced hydrogen carriers were produced for one glucose molecule. And we know there are 12 hydrogens in a glucose molecule. So 2 were added to reduce NAD, 2 were added to reduce NAD in glycolysis and link reaction respectively. 6 reduced NAD were produced in Krebs cycle and 2 reduced FAD were also produced in Krebs cycle. So this all adds up to 12 hydrogens. So what is left of glucose now? The entire molecule of glucose is stripped now. All that's left are hydrogens which are added to reduce NAD and reduce FAD. So this is what where I end the discussion of Krebs cycle. That's it from my side. Thank you so much.